All right, let's talk about the cell cycle, which also includes mitosis, and we'll talk a bit about chromosome structure as well. So if you remember back from cell theory, part of cell theory is that all cells come from the division of other cells. We don't have new cells appearing right now on Earth. So for many cells, that division of one cell into two cells is how they reproduce. Like, for example, amoeba. They don't have another type of reproduction. For large multicellular organisms like plants or animals, this kind of cell division is how they grow from their original single-celled form. Like for humans, that would be from the original fertilized egg. This is how your tissues grow. This is also how you repair and replenish cells that have died or been infected by viruses or something. So, and we call this cell division is part of the cell cycle. So the cell cycle includes everything cells do. Dividing into two cells is only part of that. So I have a couple of videos to show here that I'm going to leave in here and show you because they don't have sound, or the sound is unnecessary. So this next video that I'm going to show you is of cancer cells dividing. Cancer cells are a popular method for studying, or a popular subject for studying cell division because they divide a lot. So it gives you something to study. So, and I'm actually going to slow this down. So when you look at this, what you're going to see is some very brightly lit structures. So the way that they made this video is they used a fluorescent tag to stick to only the microtubules. Now, if you remember from our cell structures section, microtubules are one of the parts of the cytoskeleton. Microtubules are involved with several processes in the cell. Like, remember, these are microtubules are the tracks that move vesicles around in the cytoplasm. The other thing they do is move the chromosomes during cell division or mitosis. So what you're going to see when you see a cell that is not dividing, you can see the dark nucleus. Let's see. Oh, no, I can't do that right now. You're going to see the dark nucleus, and around it is a kind of diffusely lit cell cytoplasm. As the cell gets ready to divide, you will suddenly see these bright filaments. Those are the microtubules with their fluorescent tag. So anything that's dark, like the nucleus, does not have any microtubules in it. And where you see brightly lit regions, that's the microtubule. So I'm actually going to slow this video down so that it's easier to follow. Okay, so I'm slowing it down to 1/4 speed so you can see brightly lit regions. Let's see if we can pause one here in process. Okay, so notice down here at the bottom, we can see what looks like this brightly lit ball. That's the microtubules as they attach to the chromosomes and get ready to pull them apart during cell division. And you'll notice that some of these kind of get stuck, and you see this brightly lit ball that kind of sits there and gets stuck there. That's because cancer cells are abnormal. So, oh, there's a nice one that's dividing right in the middle there. Some of them kind of get stuck, and that's just because cancer cells are abnormal, and so some of the cell divisions are also going to be abnormal and aren't going to complete correctly. And that's just something that's to be expected when you're growing cancer cells in culture. Oh, there's another nice one there right in the center. So you see those brightly lit microtubules, and then separation. And then you quickly see two new dark nuclei appear. Okay. All right, so here's another little video. This one is an embedded one though, but it also has no sound, so I'm just going to play it. This is a sea urchin embryo. Sea urchins are also really popular for studying both cell division and early embryonic development because they're transparent. So you'll notice that. So this is those little tiny dots swarming around, those are the sperm. And the very large circles, those are the eggs. So here we go. Here's our first division of our embryo, and nothing is labeled here. Nothing is stained. There's no fluorescent dye. This is just using natural light. And of course, this is time lapse. Each on there, now we skip to a later time here where it's a hollow ball of cells. Now we have the formation of the different tissue layers with the internal layers forming that are going to form the gut and the muscles and the skeleton of our sea urchin. There's the primitive gut forming. So in eukaryotic cells, remember eukaryotic cells are cells that have a nucleus. In eukaryotic cells, we call this cell division mitosis. Mitosis involves the equal division of the DNA and the chromosomes, and the daughter cells are going to be identical. This is the normal type of cell division that's used for tissue growth during animal and plant growth. Mitosis is the type of division that's being used, and also for asexual reproduction. For prokaryotes, so prokaryotes are the bacteria in the archaea. We don't call it mitosis when they divide. Prokaryotes have a simpler type of a cell cycle, and this would be the same for mitochondria and chloroplasts. They also divide this way. We call the kind of division that, hold on here, there we go. We call this kind of division binary fission, meaning this just simply means split in two. And we don't use the term mitosis. We've reserved that term for cells that have a nucleus. So in binary fission, the circular chromosome replicates. It's pulled to either end of the cell, and then the cell divides. And you end up with identical daughter cells that have identical copies of the chromosome, except for new mutations, of course. And then this cell will grow larger again before it replicates. The chromosome has a single, what we call origin of replication, and that's where DNA synthesis begins, and the chromosome begins the process of replication. Now, generally, prokaryotic cells have a single chromosome, although there are exceptions to this. Bacteria and archaea as well also can have tiny little chromosomes that we call plasmids, and if you're going to go on to take micro, you may get to do some experiments with transforming bacteria with plasmids. Usually plasmids only contain 1, 2, 3, 4 genes, not very many. And also the chromosomes that are found in mitochondria and chloroplasts as well, they contain very few genes, and there tends to be more than one of them in each mitochondria. Each mitochondria might have several of these chromosomes, not just one. Eukaryotic cells, it's necessarily more complicated because we have multiple chromosomes. There's very, very few eukaryotes that only have a single chromosome, although there are some protists that only have a single chromosome. Most eukaryotes have more than that. You might know how many chromosomes human cells have. Do you know? It's 46 for a normal human cell. So that's a lot of chromosomes. And what has to happen during eukaryotic cell division? After all those chromosomes are replicated, they all have to be separated equally so that each daughter cell is going to get one copy of all of the chromosomes. So we end up with two identical daughter cells. The nuclear membrane has to break down and then reform because the chromosomes are inside the nuclear membrane, and the nuclear membrane blocks microtubules. So normally you would never have microtubules inside the nucleus to attach to the chromosomes. It would be bad to have something in the nucleus that could attach to chromosomes and pull on them. So normally the nuclear membrane keeps that from happening. So the nuclear membrane has to break down, and then at the end of mitosis it's going to reform. So here's a plant cell actually down here with the chromosomes condensing, and the microtubules are the pink structures around the outside there. So when cells are not dividing, this is most of the cell cycle. And we call this part of the cell cycle interphase. And when cells are not dividing, we can't see the chromosomes because they're not condensed. So here we have a plant cell, and here's the nucleus, and some chloroplasts here, and the mitochondria are the little red structures there. And we cannot see any chromosomes in here. There is a nucleolus, which we learned about. The nucleolus is not a membrane-bound structure. The nucleolus is where there's massive amounts of RNA transcription going on, and this is where the ribosomes are synthesized. But when the cell is not dividing, we call that interphase, and we just see the chromatin in the nucleus as kind of diffuse material. Here's an animal cell, just the nucleus here is visible, and the nucleolus. This is a really nice image, a nice TEM image. You can actually see the nucleolus with its kind of thready, ropey structure inside of there. And there's some denser regions and some lighter regions, but all of that DNA and its associated proteins that we call chromatin, that's what chromatin is. Chromatin is the DNA plus the protein that helps keep it organized. That's what we see in the nucleus during interphase when the cell is not dividing. So here's our entire cell cycle. So you notice mitosis is a small part of this. So the rest, the whole rest of the cell cycle here, we call interphase. And interphase is divided up into three phases, which we call G1, S, and G2. So G1, G is for gap, because early scientists were studying the cell cycle, thought that this was a gap in activity of the chromosomes because they could see that DNA synthesis was happening at a different time, so we call that S. This is when all the chromosomes are being replicated, getting ready for cell division, and then G2, this is another gap. You could also think of G as being for growth, because we now know there's an enormous amount of activity going on during G1 and G2. Gene transcription is taking place during this time. Cell metabolic activities are taking place during this time. It's not like nothing is happening. But this is when replication of the chromosomes occurs during S, and then mitosis. And you'll notice we have a second word here, cytokinesis. So this is what we refer to as the separation of the cytoplasm and the membrane. So everything that's not the nuclear material, we call that cytokinesis. So if we had labs, we were going to look at some dividing cells. This happens to be an onion root tip. That's one of the slides we were going to look at in lab. And you can tell when cells are getting ready to divide. Now, if you notice here, you can see the really rectangular-ish plant cells here. Now, the ones that are clearly not dividing, like this one right here, we can see the nucleolus. We can see that kind of diffuse chromatin material. We cannot see any distinct chromosomes in this image. So this cell is clearly an interphase. Now, the first stage, as these cells start to get ready to divide, look at this one right here. So we have, you can still see that there's a nuclear membrane there. You can see the outline. And we can kind of see the nucleolus still there, but it's starting to break up. But we can now see these thready chromosomes. So the chromosomes are starting to condense and become really visible as distinct chromosomes. This cell down here is at a slightly later stage, and we can see that the nuclear membrane is gone here. We don't see that nice boundary. The nucleolus is gone, and we see much denser chromosomes as they continue to condense. So here's a nice image where most of the cell has been digested away, and all that's left are the microtubules and the chromosomes. So we can see our nice duplicated chromosomes. And if you look closely at these chromosomes here, you can see that they're made of two strands that are lying side by side there. And then we have our microtubules, which are attached to the chromosomes. And you can see why we call the ends of this spindle. Okay, so this structure here, the microtubules, as they attach to the chromosomes, this is called the spindle, or the mitotic spindle. And then the ends of the spindle are called the asters. And aster means star, and you can see why they would be called that, because they sort of look like a star. And you might wonder why this is called a spindle. This is another one of those old-fashioned words that has gotten frozen in biology naming. There's a lot of them. And then when this word was coined over 100 years ago, everybody knew what a spindle was because everybody had one in their house. A spindle is found on a spinning wheel that's used to take raw wool or cotton and spin it into yarn. And it's what the yarn wraps around. So it's a sharp peg that sticks up, and then the yarn wraps around the spindle, and it's generally fatter in the middle and thinner on the ends, and that's why this looks like a spindle full of yarn. So we still call it a spindle today. All right, so before cell division, DNA already has been replicated, and what we can see here in this scanning electron microscope image of a chromosome is the two replicated strands of the DNA. So it's all wound around proteins though, and that's what actually makes up these furry chromosomes here. If you took the piece of DNA from a human chromosome and stretched it out, it would actually be 2 to 5 centimeters in length. That's about that long. So that's a lot of DNA. So it's all wound around proteins to get to this small size here of only a couple of micrometers in length. So what we actually have here in our chromosome that's ready for mitosis is 2 strands of DNA that have been replicated, all wound around their protein. So we have one here, and we have one here. So we call these 2 identical pieces of the chromosome the sister chromatids. Sister chromatids are made by DNA replication, and they are identical. The centromere is a slightly different region of the chromosome. It's if you look at the sequence of DNA in the centromere, there's no genes there. It's a repetitive region of DNA that attracts the right proteins to bind so that microtubules can bind here during separation of the chromosomes during mitosis. But there is one continuous piece of DNA going all the way down here, all wound around protein. So after DNA replication, we have 2 copies. These 2 copies are called the sister chromatids. So here's our cell before anything happens here during replication. So we have a single chromatid, and one chromosome can be one chromatid, like here, or it can be 2 identical chromatids like we saw in the previous image here. So we have one chromatid. Our cell here has one chromosome. That chromosome has one piece of DNA in it right now. So the first thing that has to happen is DNA replication, and this actually occurs during interphase, during S phase of interphase, S for synthesis, not during mitosis itself. Okay, so now we have 2 identical sister chromatids, which means we have 2 identical copies of the DNA. And now, when the cell divides, these two identical copies are going to separate, and one is going to go to each daughter cell. So mitosis occurs, the sister chromatids separate, and now we have two daughter cells that are identical to each other, and they're identical to the mother cell. Now, calling these the mother cell and the daughter cells, that's just traditional in cell biology. It doesn't have anything to do with whether the cells are actually female or not. We still call these the two daughter cells. So daughter cells identical to each other, and the daughter cells are also identical to the original mother cell. So here we have pairs of chromosomes. So for human cells and also for other eukaryotic cells, the normal condition is what we call diploid, having two copies of each of the chromosomes. So humans have 46 chromosomes, that's 23 pairs of chromosomes. So a homologous chromosome is not identical. Homologous chromosomes have the same genes in the same order, but not necessarily the same version of that gene. So once those chromosomes replicate, now the identical sister chromatids are made by DNA replication. They have this, they're the same, but the pairs of homologous chromosomes are not identical. So the chromosome number for each species is species specific. The 10 to 50 chromosomes is the most common for eukaryotes. Human cells have 46, so we're right in there with the average for eukaryotic cells. The lowest would, of course, be one chromosome. There are a few protists that only have one chromosome, and there are some eukaryotes that have hundreds of chromosomes, and for some reason, most of those with very high chromosome numbers are plants. There are also some animals that have very high chromosome numbers, but they're much more rare. Plants, for some reason, can have huge numbers of chromosomes. It doesn't seem to cause them a problem. When we look at all the chromosomes for one individual, we call that a karyotype. So karyo, we can see the root in there again that we used to mean nucleus, as in eukaryote and prokaryote. So karyotype is what's in the nucleus of a particular individual. Now, this isn't the chromosomes from a single nucleus in one cell, because it takes a lot of smashed cells to get nice photos of all of the chromosomes in a particular organism. So a karyotype is for one particular individual. So if they were going to do a karyotype of you, they would take a blood sample, get a sampling of your cells, and then take the best image of all of your chromosomes and compile them together into a karyotype. So this particular individual had an X and a Y chromosome, so very high likelihood that this particular individual would be a male. But you can see there are pairs of all the chromosomes. They are numbered according to their size. So chromosome 1 is the largest chromosome. Chromosome 22 is the smallest one, except for the Y. It's actually the smallest one. And each chromosome has its particular assortment of genes that are present there. So a pair of chromosomes, so these would be a homologous pair, and they have the same genes, but not necessarily the same version of that gene. So all that DNA is packaged with proteins and wound around proteins. Genes, even in a bacterial cell, the DNA strand is huge. So for an E. coli, so you can see somebody did a nice artistic picture here. This is a single chromosome. You can kind of see the remnants of it there. The single circular bacterial chromosome from an E. coli, where all the protein was digested away, and that allowed all of the coiled DNA to kind of explode outwards here on the slide, and they could get this image. So even for a bacteria, their circular DNA chromosome is tightly packed and supercoiled with protein so that it can fit, because it's actually a really long piece of DNA. I mean, all these loops and loops and loops of DNA, if we stretched it out, we could stretch it out into a big circle. But that would be much larger than the size of the entire bacterial cell. So it is packaged with protein to organize it into a much smaller region. Now, our chromosomes do the same thing. So the word chromatin, which you've heard before, chromatin is what's found in the nucleus, and the chromatin is made of DNA and protein. It's actually more protein than DNA is in the chromatin, and most of that protein that's found in the chromosomes is organizing the DNA. Because of this elaborate system of packing. Now, this little illustration down here at the bottom. So here's our DNA strand wound around. These protein beads winds around twice. These proteins here are called histones, and that's the first level of organization of keeping the chromosome organized. So here is a human chromosome where the same thing was done. So you can kind of see the remnants of that X-shaped chromosome here. All the protein was digested away, allowing that DNA to spill out onto the microscope slide. And you can see this huge spaghetti network of strands. This is one piece of DNA. This is one molecule of DNA. So DNA, again, remember, is a long chain of nucleotides that we abbreviate with the A, C, G, and T, and that's what we're looking at here. This is, and it's even cut away so we can get higher resolution here of the image. This is one DNA molecule from one chromatid of a chromosome that has spilled out onto the slide. So this is an enormous amount of DNA and a hugely long piece of DNA. And we have 46 of those in every cell. And after DNA replication, there's 92 DNA molecules in the nucleus of every cell before cell division that needs to be packed and organized so that it doesn't get tangled and tear apart. So the first level of organization are those beads on a string, we call it, the nucleosomes. And here's an actual image of what that looks like. So our naked DNA molecule is wound around histone proteins, which have amino acids in them with positively charged side chains, and that allows our negatively charged DNA to stick to it. So DNA has a lot of negative charges. It has those bases, A, C, G, and T. It has oxygens in the ribose and in the phosphates that have negative charge. So it's going to stick to the positively charged histones. That's our first level of organization, and that's called the beads on a string organization. The next level of organization, these beads are packed up into a fatter fiber, and you don't have to remember the names of these fibers that just have numbers associated with them. The next level of organization is the 30 nanometer fiber, and then those 30 nanometer fibers are wound around into a fatter 300 nanometer fiber, and then these are packed into the mitotic chromosome, the fully condensed mitotic chromosome. And exactly how we go from this organization to the metaphase chromosome is not completely understood. The proteins involved are known, but exactly how this is packed into there is not completely understood yet. But this is what keeps that huge, long DNA molecule organized in each of these sister chromatids. So the phases of mitosis. Humans like to organize things, and to the cell, all of these cell divisions and phases are just blend continuously one into the other, and We like to define particular phases, though, and so we can talk about all the different things that happen during mitosis. But keep in mind, there's no abrupt end to each of these phases. And one side of the nucleus might be in one phase, and the other side of the nucleus might actually be in the next phase. And that's because to the cell, this is one long process. We just kind of artificially divide it into phases for our own use. Okay, so we're still in interphase here in the first one, and we could tell that this cell is an interphase because we can't see any defined chromosomes here. If we were, and I'll show you some actual microscope images of cells shortly, we don't see any chromosomes. All we see in the nucleus is diffuse material. When you're looking under the microscope, it often looks like sand in the nucleus. And you might see a nucleolus as well. That's another indication that transcription is occurring, metabolic activities are occurring. So at the end of interphase, you remember the phases of interphase were G1, S for synthesis, DNA is replicated, and then G2. So in G2 here of interphase, that means that DNA replication has already occurred. We have 2 copies of each chromosome, even though they are not condensed yet. The centrosomes, for species that have those, are going to replicate. Remember, centrosomes are pairs of centrioles. They're made out of microtubules. They are the microtubule organizing center of the cell for cells that have them. So most plants and fungi don't have them, but animal cells do. So human cells definitely have these. So in the end of interphase, the centrosomes replicate and start to migrate to opposite sides of the nucleus. Once the mitosis has actually begun, the earliest stage of mitosis we call prophase. And depending on what source you're looking at, sometimes these 2 phases together are all just called prophase. And sometimes they're called early prophase and late prophase. Your textbook calls them prophase and prometaphase. But it's because prophase is very long and a lot of stuff happens during prophase. So it's useful to divide it up into 2 different phases, an early and a late. The first things that happen during prophase are the separation of the microtubule organizing centers. And they separate because these microtubules are pushing away from each other and lengthening. These microtubules are actively being synthesized and lengthening and walking apart from each other and pushing those microtubule organizing centers to opposite sides of the nucleus. The first thing that will be visible to us under the microscope will be the condensation of the chromosomes. You'll start to see the nucleus of the cell will go from being just looking like sand to being thready spaghetti strands in the nucleus. Then in late prophase, or prometaphase as it's often called, so prophase again is broken into 2 phases. We could call this whole entire thing prophase, or we could say early prophase, late prophase. Your textbook says prophase, prometaphase. So in the later parts of prophase, the nuclear membrane breaks down, and this will also be visible looking at specimens under the microscope. And now, when the nuclear membrane is broken up, now microtubules can finally extend into that space, and they will attach to the chromosomes and get ready for the next step. So microtubules will attach to both sides of the chromosome at the centromere, wherever that is. So the centromere isn't necessarily in the middle of the chromosome as it's kind of depicted here. It could be at either end or anywhere along there. So both sides of the centromere will be attached to a microtubule, and then these will start pushing and pulling on the chromosomes until they get to metaphase. So what we call metaphase is when we finally see all of the chromosomes lined up along this imaginary plane. We call the metaphase plate. And in this cartoony depiction here, it looks like all the chromosome arms are also lined up, but in fact, the arms are not attached to the microtubules. Only the centromere is attached to the microtubule. So the chromosome arms are flopping all over the place, and I'll show you some actual microscope images in a second that'll show that. Okay, so now at metaphase, we have the centromeres of the chromosomes all lined up, attached. Each sister chromatid is attached to a microtubule via the kinetochore. The kinetochore is a complex of proteins that attaches at the centromere when the cell is getting ready to divide. The next phase, anaphase, and again, these are described, the names of the stages by what was initially seen under the microscope. So these were visibly different stages to the early microscopists. So ana means, it's from the Greek root that means separate or apart. So this is when we see the two sister chromatids start to separate. And now we consider them, as soon as they separate, those two sister chromatids, now we consider them two separate chromosomes. So back here in metaphase, these chromosomes are still considered one single chromosome with two chromatids. Once they separate, those two chromatids are now considered single chromosomes. So now we have chromosomes that have single chromatids. All right, so the chromosomes move towards the ends of the spindle, and finally the last stage, telophase, we have the reforming of the nuclear membrane and the chromosomes start to decondense, also something that will be visible under the microscope. You'll be able to clearly see, especially in the onion root-tipped cells, that the nuclear membrane is reforming. Now, there's a second word here, cytokinesis, and this is often how it's depicted in textbooks as telophase and cytokinesis, but actually these describe different processes. Telophase is describing what's happening to the nucleus. The nuclear membrane is reforming. Cytokinesis is a description of what's happening to the rest of the cell. So we have the cytoplasm has to separate, the nuclear, or the cell membrane, the plasma membrane has to separate and separate these two cells. And that's what cytokinesis is. And we can see in the root here, cyto means cell, kinesis is move. So cytokinesis is the separation of the rest of the cell, not just the chromosomes of the cell. So this is a little video, also no sound, so I'm just going to play it for you here. And this is two different light images of the exact same thing. So on the right here, we have just light, just high contrast regular lighting. And you can see that, well, this is kind of a freeze frame in the middle of the movie. But what we're going to see is shortly after fertilization, we have two nuclei. We have the egg nucleus and the sperm nucleus are going to fuse. And then this, I believe this is a sea urchin embryo. Again, we like sea urchin embryos because they're transparent and easy to get in large numbers. So we're going to see the two pronuclei fuse and then two cell divisions. And because nothing is labeled over here, we're just using regular light, just high contrast lighting. You can see the nucleus here by the absence of the cytoplasmic material. And you'll see the nucleus disappear and reform. On the left side here, what is labeled is the nuclear membrane is labeled with a fluorescent tag. So this is the exact same movie, just viewed one with regular visible light, high contrast lighting, and the other with specifically for this fluorescent tag, the wavelength that is being given off by the fluorescence in this particular one. So you'll see the nuclear membranes disappear, and while on the right side here, that's when the cell is dividing, and then you'll see the nuclear membranes reform. So it's kind of short, so we'll watch it a couple of times here. All right. So here's the two pronuclei fusing, the egg and the sperm nucleus. Now it's going straight into the first division. The nuclear membrane is gone. And now here's our two new cells forming. and now their second cell division is starting. We're in prophase of the second cell division. And now we have an embryo with 4 cells. Okay, let's watch that one more time because it's kind of short. And it's hard to attend to both sides at the same time. So let's watch it one more time. All right, so here's our 2 pronuclei. The egg and the sperm nucleus are fusing, each with half the chromosomes for the new organism. Okay, first cell division. Now here's telophase of the first cell division. We can tell it's telophase because the nuclear membranes reappear. Now it's going right into the next division, prophase of the next division. I can tell it's prophase because the nuclear membrane is breaking down. First one cell, then the other cell, and now it's telophase. Nuclear membranes reappear. Okay, so here's some microscope images, and in these now we have 3 different fluorescent tags going on in these images. So let's talk about these. Wait, I need my pointer back. Okay, so we have 3 different colors of fluorescent tags here. The chromosomes are labeled with a blue fluorescent tag, so every place the chromosomes are, we can see blue. We have microtubules that are labeled with a yellow fluorescent tag, and we have intermediate filaments that are labeled with an orange fluorescent tag here. So we can see very clearly here. So now let's look at G2 of interphase. So G2 is after S, so that means DNA has already replicated, and so each chromosome here, even though we can't see them, each chromosome has 2 sister chromatids that are identical because they were made by DNA replication. We have our microtubule organizing center in the cell here, and you can see the yellow microtubules radiating out from there. So the first thing that's going to happen here for the cell to divide after DNA synthesis is these centrioles have to replicate in the microtubule organizing center, if it's an organism that has those, because, you know, remember that most plants and fungi don't have centrioles. But the centrioles are going to replicate and start to separate. So here we can tell it's prophase. The first thing that we can see here is that the chromosomes are starting to become thready. They've gone from being kind of solid looking to looking kind of like spaghetti. But we can see that the nuclear membrane is still intact. So this is early prophase, or what your book calls just prophase. And now we can see those centrioles starting to separate and move. You can see the 2 little bright hot spots there next to the nucleus. All right, later in prophase, what we're going to call prometaphase, we have now these 2 little bright hot spots here are completely on opposite sides of the nucleus. The nuclear membrane is gone, and we can tell that it's gone because now we can see the arms of the chromosomes kind of sticking out here all over the place. And you can see these microtubules extending now into the nuclear space, which they were not doing before. For metaphase, we now have a very bright spindle, very dense formation of microtubules. And because this yellow fluorescent tag is staining the microtubules, it's going to look really bright as there's more microtubules present. And you can see the ends of the spindle here. This is when it looks most like an actual yarn spindle, is at metaphase. And see here how the chromosome arms are kind of going in all directions. The chromosome arms are not attached. So on each chromosome, again, we have the centromere, which could be anywhere along the chromosome, and then 2 arms on either side of the centromere. And the arms are not attached, so they're kind of going all over the place. But the centromeres are lined up here on the metaphase plate. For anaphase, and again, ana means separation. So this is when we can see those chromosomes have now separated. The identical sister chromatids have separated, and the centromeres are being pulled towards the ends of the spindle. Telophase, we can still see microtubules here. So what are these microtubules doing? The ones that are here, they're obviously not the ones pulling on the chromosomes, because the chromosomes have already made it all the way to the end here. So these microtubules are walking apart from each other and pushing those ends of the spindle further apart before cytokinesis is going to occur. Okay, so telophase, that is the reformation of the membranes, and cytokinesis, that's the separation of the rest of the cell. And it occurs differently in plants and animals, and we'll talk about the difference between that. And why do we separate these? These are not the same thing. Why do we have telophase and cytokinesis? It's because sometimes mitosis occurs and cytokinesis does not occur. There are certain organisms, some protists, and even fungi that divide and have multi-nuclei in what seems to be a single cell because cytokinesis does not occur. That happens during fruit fly embryonic development as well. The first rounds of mitosis occur without cytokinesis, and you get a large syncytial cytoplasm with many nuclei and no cell divisions, and then the cells divide later. So let's take a break here, and I'm going to stop it, and I'm going to link to these two videos here. The first one actually I think has no sound, so let's watch that one, and then we'll stop so you can go watch the other one. All right. So the first one here, this is, hold on here, I'm going to slow this one down. So what we have here is a little piece of a Drosophila embryo. Drosophila embryos, Drosophila is a fruit fly. Drosophila are kind of jelly bean shaped, very tiny, tiny little jelly beans, smaller than a grain of rice initially, maybe half the size of a grain of rice. And this is just the little, let me go, oh, I don't have my pointer anymore. This is just the little end piece of that. And what's labeled here is microtubules. The microtubules are the only thing that's visible. They're the only thing that's lit. So what you're going to see, and they're labeled with a yellowish fluorescent label. So the dark circles that you see, those are the nuclei, and that tells us that the nuclear membrane here is still intact because the dogs are seeing another dog apparently outside. So the reason that the nuclei all look dark is because the nuclear membrane excludes microtubules. Microtubules cannot enter the nuclear space, which is a good thing because we don't want something in the nucleus that can attach to the chromosomes and pull on them. That would be bad. So that's why the nuclei look dark in this image. So what you're going to see is bright hotspots develop as the microtubule organizing centers start to synthesize or start to project more microtubules. Then you're going to see the nuclei disappear and a bright spindle is going to form. And then you'll see those spindles lengthen and then new nuclei are going to form. All right. So here we go. And you'll notice that the wave of division is not exactly simultaneous across the entire embryo. It kind of starts on one side and moves down. So nuclear membranes are breaking down. We can tell because we now see microtubules in that space and we don't see those dark circles. We can't see the chromosomes because they are not labeled in this preparation. We can only see the spindles. All that's labeled is microtubules. So here are the, we're getting to metaphase. Once you start to see the ends of those spindles push away from each other, that means we're getting into telophase. See those spindles lengthening, that's telophase. So now you can see the new nuclear membranes are forming. There, and so now we had one round of mitosis without cytokinesis. All right. All right, so this next video here actually has sound, and so that doesn't play very well. It has music and stuff. So I'm going to stop the video and put this video in the link for the order. Watch that and then come back for the next part.